Okay, so last couple of things I want to talk about with respect to epigenetics. Really interesting parts. So one question. This idea that they can be dynamic and that they can respond to environmental influences. So with respect to us, do they change over time? Yeah. Does our epigenetics change in our somatic cells as time goes by? And there's good evidence developing that suggests the answer is yes. So your epigenetics now is not the same as your epigenetics when you were 2 years old, or 10 years old, or 15 years old. Or in my case, what am I now, 32? So when I was 31. So twin studies, again, have been really interesting with respect to this. Here was a paper published a while back in PNAs, epigenetic differences during the lifetime of monozygotic twins. So their chromosomes started off genetically at least identical in time. So this was from this paper, and I took it verbatim because I thought it was a really nice statement. We found that although twins are epigenetically indistinguishable during the early years of life, in other words, the two identical twins were the same in terms of their epigenetics when they first started off, older monozygotic twins exhibited remarkable differences in their overall content and genomic distribution of 5-methylcytosine and histone acetylation, affecting their gene expression portrait. And this was some of the data that they showed. Here is methylcytosine. So methylation of cytosines on the DNA, which in the last hour we talked about that. 3-year-old twins here, 50-year-old twins. One of the things we see is a significant increase in methylation and divergence, where the values are different between the two twins. And then here is acetylation on histone H4. 3-year-old twins, very similar. 50-year-old twins, markedly different. And then here, acetylation on histone H3, very high in 3-year-old twins and very similar, and then decreasing as the person gets older. And again, divergent as time goes by. So we are all divergent, if you were wondering if that was the case, if you've ever seen that movie. If you don't know what I'm talking about, don't worry about it. So comparative genomic hybridization for methylated DNA. These are kind of neat studies as well. Yellow, if the chromosome appears yellow, that is very similar pattern of methylation between the two chromosomes. If it is red, it's hypomethylation in A relative to B. And if it is green, it's the other way around. Hypermethylation of A relative to B. And so here are 3-year-old twins. We're essentially looking at the entire chromosome and saying, what kind of differences do we see in methylation between the two twins? And the more yellow you see, the more similar is the pattern of methylation. So 3-year-old twins, lots of yellow in terms of those two chromosomes. And then this is looking at 50-year-old twins. We see lots of red, and we see lots of green, suggesting that there's differences, major differences that have accumulated in time as a person has gotten older. What does that mean with respect to diseases? And that's a really good question. And at this point in time, for a lot of this, we don't know for sure. Is it contributing to diseases? Probably yes, in terms of these changes. So some patterns of methylation may be beneficial and reduce our risk. Others are increasing our risk as time goes by. I put this in here as well. Here was heritability data for schizophrenia. And one thing we saw, this is the concordance between monozygotic twins for schizophrenia. It's 0.47. Two identical twins. Right? They are identical at the genetic level. If one of them has schizophrenia, there's only about a 47% chance that the other twin will also have schizophrenia, which is rather low. Now, the heritability in terms of the variation in the population is still pretty high. It's 0.7. But a good question is, why is the concordance low? When is schizophrenia typically diagnosed? It's older population. You know, a typical person is not going to be diagnosed at 8 years of age or 10 years of age. 20s, 30s, 40s, around there. So there has there's been time for epigenetic divergence between identical twins. This could very significantly contribute to this low concordance for schizophrenia. So diseases that are later age of onset, that are multifactorial, are more likely to be influenced, I think, by epigenetic divergence as time has gone by. And that has then kind of changed this, because this epigenetic divergence is probably more, certainly has a greater frequency of randomness that occurs. So they started off genetically identical to each other, but epigenetically, they diverged as time goes by, is the fundamental idea. So if our epigenome changes, what kind of things induce these changes? You know, the identical twins go off and have different lives. Those different lives influence what kind of epigenetic changes occur in their DNA. So I give you a whole bunch of data. I'm going to skip through some of it because, one, I'm not going to test you on it, but there's some interesting things, so I'll just do it really quickly. This is kind of not the nicest study in the world, but it's fear conditioning, where a rat will get a shock in the cage under certain conditions. And that's called fear conditioning. Fear conditioning induces changes in DNA methylation in the brain, and in particular regions, in the brain-derived neurotropic factor gene promoter. So those who have been exposed to this fear conditioning and those who have not show differences in their epigenetics for that particular gene in their brains. Social deprivation in rats during the first postnatal week causes changes in DNA methylation in the same gene in the brain as well. So our genes responding with epigenetic changes in response to the environment. So could this be seen in humans as well? It's entirely possible. Decreased expression of this gene in the prefrontal cortex. A schizophrenia-like state can be induced in mice by giving them a diet that is high in L-methionine. And L-methionine is the substrate for methyltransferase enzymes. So methyltransferase enzymes, those that methylate CpG ions. Give them a high concentration of the substrate, you can induce a schizophrenic state, implying that it is these epigenetic changes that are resulting in that. Cocaine exposure. There's lots of studies on cocaine exposure as well. So, quick summary of this. Epigenetic traits, heritable phenotypes, changes in the chromosomes that don't involve changes in the sequences. Lots of interest in this. Methylation of the DNA and histone acetylation of the DNA. Errors can occur, resulting in different disorders, including cancer. Epigenome changes as time goes by. And the divergence of the epigenome could also be a part of how these diseases present. So if you recall back in my first objective, I said the two things that influence a multifactorial disease, genes and environment. The third one would really be epigenetics as well. So genes affect it, environment affects it, and epigenetics affects it. And epigenetics may be a manifestation of the environment. So you could probably lump epigenetics in with environment. Sorry about that. So any questions before I move on to my next? It's nice that I have 3 days, so even if I get behind, which, by the way, I always do, never fails, then I can make sure I get everything covered. Okay. The last thing that we want to cover today is genomics and personalized medicine, which is also, I think, a really interesting area as well in terms of new discoveries being made almost all the time. So these are the objectives. And I don't know if there's anything that I want to go into detail in terms of those objectives. Hopefully they're pretty straightforward. You guys have access to Clinical Key, correct? Yeah. So there's a couple of books in genetics that I particularly like in Clinical Key. There's Medical Genetics by Jordi. That one is kind of the one that I use a lot. So if you're wanting to go to a textbook, I would recommend this one. Another one is Thompson and Thompson, Genetics and Medicine. This one is a little bit higher level. So if you've read through this and aren't making sense, the medical genetics kind of simplifies it a little bit more. But if you're looking for the high level stuff, Thompson and Thompson is a little bit better. So genomics. When we say genomics, what are we talking about? And essentially what we're saying is the study of the complete set of genetic instructions contained within an organism is genomics. So sequencing, getting the entire sequence. So how many different species have been at least partially sequenced? Surprisingly, this is a really hard thing to find. I've done this search pretty much every year when I've done this, and I've had a hard time finding it. But this gives you some idea, lots. And you don't have to memorize this table or anything like that, but just realize that there's a lot of different species that have now been fully sequenced, especially in terms of bacteria. In the humans, at least 2,500. And every year I update this. Last year it was 1,092. This year it's about 2,500 individual human genomes fully sequenced. The UK has recently started a project that they're calling the 100,000 Genome Project, where within a certain amount of time, and I forget how much time, maybe 10 years from now, they would like to have an additional 100,000 genomes in terms of library. And I think they'll make it easily. The $1,000 human genome is already here. This is a company, and just so you know, I have no stock whatsoever in this company, but they are producing this system, and they say, you buy our system, you will be able to do a genome for $1,000. Now, you can charge $1,000 per genome. The cost of this setup is hundreds of thousands of dollars to be able to do that. But once you have it in place, you should be able to do the $1,000 genome. This was taking it out to about 2013 or so. This is a more up-to-date figure for this in terms of the cost per genome. And you can see right around 2014, 2015, there was another big drop in terms of us becoming more and more efficient with respect to genome sequencing. If you have a test done, let's say you need an MRI. How much does an MRI cost? I don't even know. I have no idea. Hundreds of dollars, right, to get an MRI. Maybe close to $1,000. So sequencing your entire genome is now, not in a few years, but now reasonable to do. Is it wise to do? That's a different question entirely. Sometimes yes, maybe sometimes no. But by the time you guys are out there, this is going to become more and more and more prevalent, where people will have their genome, or at least a large percentage of it, where they already have that information in hand. So, questions. Why would we want to do this? Why sequence? One, improve health and advance the treatment of human diseases. When the Human Genome Project was first proposed back in the 80s, this was the driving force, is to improve health and advance the treatment of human diseases. So how? One is an understanding and identification of every single gene in the human genome. A second part is, how are these genes regulated? When are they expressed? How are they expressed? And to what level are they expressed? The analogy that I like to use a lot is identification of all the genes within a genome is essentially assembling the library. Or sorry, no, assembling the dictionary in the library. So the dictionary just has all the words. So identifying all the genes is just simply coming up with all the words in the dictionary. Each and every cell is like a novel. It is how all of those different genes are expressed. So that's the next step, is how is a neuron expressing its genes compared to a skeletal muscle cell, and how is it expressing its genes? So cell type specific gene expression profiles. How are all of the different genes that we have expressed to make a neuron different than a skeletal muscle cell? Variations within a population and the significance of those variations. So I showed you genome-wide association studies. That is where we're looking at genetic variation in a population to try to identify genes that are associated with disease, especially multifactorial. Linkage of genetic variations with phenotypes, improving our ability to do prognosis. Based on this particular mutation, you have a certain probability of having this particular disease. And then treatments, developing tailored treatments that are specific to your genetic information. And that is the domain of pharmacogenomics. Using genomics to improve, essentially, drugs. Personalized medicine is what it amounts to. Non-human genomes. One, increase our understanding of life on Earth. There was a study that Craig Ventnor did a few years ago. And Craig Ventnor was one of the major guys with respect to the Human Genome Project. And he has his own research lab now. He's a bit of a blowhard, but he's very passionate about what he does. One of the things he did is he took some ships and went out into the middle of the Atlantic Ocean and took these 55-gallon drums and went to a whole bunch of different locations and just picked up water from the ocean, brought it back to the lab, and sequenced everything that was in there. So all of the different bacteria. And from those studies, identified so many different types of bacteria and viruses that we had never characterized before. Just by going out in the ocean, getting these 55-gallon drums of water, and then sequencing everything that's there. It's really interesting stuff. What is this? Anybody know? Ebola. Yep, this is Ebola. So when there are new outbreaks of diseases, one of the very first things that happens now is sequencing of that microorganism. We saw it with Zika as well. Within weeks, they had the complete sequence of the Zika virus that was causing the rise in infection rates in places like Brazil, for example. Why? Why would that be beneficial? One is it different? Is there a difference in this Ebola and this outbreak compared to an Ebola outbreak that was 10 years ago, for example? And if so, does that tell us something in terms of how we might be able to treat that particular infection? So, very rapidly, identifying these changes. So the genes and the gene expression of pathogens. The evolution of new strategies or new strains of these pathogens, and then new strategies as well to develop new pharmacological treatments that target those particular changes, trying to understand the differences. Understanding human microbe relationships, for instance. Lots of studies not too long ago published in terms of our microbiome, for example, from our gut. And the sequencing of all of the different microorganisms that are present within our microbiome. What's there? How does it change over time? If you have a particular infection, how does your microbiome change? Genetics and sequencing can allow for that. Identification of highly conserved DNA sequences. We can compare various species. And those sequences that are highly conserved are probably more important sequences as well. So if we see yeast and humans have a certain sequence of DNA that has hardly changed over evolutionary time, it suggests that there may be something important about that. To elucidate what makes us human. Interesting studies as well, looking at, for instance, we have, shocking here, we have the ability to speak, right? Yep. Chimpanzees don't. So, our closest relative. What's the difference between us and chimpanzees in terms of our ability to speak and their lack? What has happened? And so there's some really interesting studies there as well in terms of what are the genes that are contributing to that. So the human genome, haploid genome is about 3.2 times 10 to the 9th base pair, 3.2 billion base pair. So a diploid cell has about 6.4 billion base pair of DNA. That is what we call our C value. There's a C value and then there's an N value. The N value is the number of chromosomes. So N is number. C value is just DNA. So our C value is 3.2 times 10 to the 9th base pair. This corresponds to approximately 20 to 25,000 total genes within our genome. If depending on how you define genes. An apple tree, in comparison, has about 57,000 genes. That can be a little bit humbling for some people. That an apple has twice as many genes as a human being. A fruit fly has about 14,000 genes. So we can feel good that we have more genes than Drosophila. But I don't think anyone would argue that we are more complex, especially neurologically, than an apple tree or a fruit fly. So a good question, too, is how do we have so much complexity with a relatively small number of genes? And the answer seems to be we're a lot more creative in terms of how we express these genes and how we splice these genes as well. Alternative splicing is much, much higher in humans than it is in just about any other species. We can make more variety of proteins from the rather small number of genes that we have. About 1.2% of the human genome actually codes for proteins. So the vast majority is not protein coding. And there's lots of debate in terms of what the rest of it does, if it does anything. I'm not going to get into those details at this point in time. If you look at genome size, you see some interesting things as well. Not just genes, but how much DNA is present there. And a couple of species tend to stand out. Salamanders really stand out, and lily really stand out in terms of the amount of DNA. Huge amounts of DNA. Here we are, essentially on order with the toads. Yeah. We have about the same amount of DNA as a toad does, a little bit more than a chicken. So genome size is also not a good indicator of the number of genes either. So if you look at E. coli, for example, 88% protein coding, 4,200 genes, about 4.6 million base pair. Drosophila, 180 million base pair, 14,000 genes, about 10% protein coding. And then you get to us with about 1.2% protein coding and somewhere around 20 to 25,000 genes and 3.2 billion base pair of DNA. So lily doesn't have a lot more genes than an apple tree, even though it has a lot more genetic material. Why does it have that much more is a good question, but we're not going to go into that today. Next thing I want to talk about is personal genomic profiling. And this statement here, which is entirely my own personal view, so you can take it as you want. But if you think patients won't be bringing this kind of information in, I think you ought to rethink that opinion. In 10 years' time, they will most definitely, I mean, even now, they will be bringing in their own personal genetic information and their own personal research on this genetic information, which may not necessarily be accurate. And they will be bringing it to you and saying, what do you think? I've identified that I have this particular polymorphism and this particular polymorphism and this particular polymorphism. And I expect you to be able to tell me what that means. And you might have to say, I'll get back to you on that. But this was what 23andMe, probably most of you may be familiar with 23andMe. They do a lot of personal genomic kind of profiling. They had this on their website. For $99, you could get reports on 240 plus health conditions and traits, and a lot of other stuff. This was a few years back. So in 2013, the FDA sent them a letter. And this is not the kind of letter you ever want to get from the FDA. And the FDA said, essentially, Dear 23andMe, we're sending you this because you are marketing this saliva collection kit and personal genomic services without marketing clearance or approval in violation of the FDA Act. You were in violation of the Food and Drug Administration. Companies that do this kind of work, or pharmaceutical companies, do not like to get letters like this. This can be really, really damaging. So this was soon after the warning letter. Find out what your DNA says about you and your family. No mention at all about genetic diseases. Cost was the same, but no mention of diseases. Here is what it is now, because things have changed in the last few years. So there's carrier status report, 35 different reports. So they are now testing and they will give you this information with respect to cystic fibrosis, sickle cell, hereditary hearing loss, ancestry reports, wellness reports in terms of lactose intolerance, for example, and then trait reports. I love this one, unibrow. They will tell you what is your likelihood, perhaps, of having an offspring with unibrow. Okay. Hair loss, yeah. The ability to taste sweet versus salty in terms of traits. This one you don't have in your handout, and I forgot. You don't have this one, right? I was going to put this in, and then I forgot. Is that in April of this year, they got additional FDA approval to test for a number of different genetic diseases. So 23andMe can now provide a report for factor V Leiden, alpha-1 antitrypsin, late onset Alzheimer's, looking at ApoE4, Parkinson, looking for 2 different genes, Gaucher disease, factor 11 deficiency, celiac, G6PD deficiency. What does G6PD stand for? Oh, okay. You've had your biochemistry, I can tell already, right? Yeah. What pathway is it? Pentose phosphate pathway, right? Resulting in a decreased expression of NAD, pH. Yeah, all over it. Okay. Dystonia, and then hereditary hemochromatosis. Hemochromatosis is a particularly interesting one. What's the mode of inheritance of hemochromatosis, hereditary hemochromatosis? No? It is an autosomal recessive disorder. Hereditary hemochromatosis has reduced penetrance. There are 2 major alleles for hemochromatosis, and I forget the exact designations for them. They're both in the HFE gene. One of them results in, if a person is homozygous, remember it's autosomal recessive, if a person is homozygous for HFE gene mutation, they have about a 20% chance of actually developing hemochromatosis. So think about that in terms of you as a primary care physician. A person comes in, they have had this genetic profiling done. 23andMe has told them that they are homozygous for an HFE mutation. As far as they are concerned, they have hemochromatosis, probably. Do they? How old are they? Hemochromatosis is something that doesn't develop until typically you reach the age of about 40, 50, 60 years of age. And it's different males versus females. Males much more likely to develop than females. So there's a lot of complexity associated with hemochromatosis. It's not as simple as positive versus negative. So is this problematic? And I would say that it is, in many respects, problematic. Because people are going to be getting a lot of genetic information that they don't necessarily understand. Does 23andMe provide information? Yes, they do. Is there somewhere in their information where they explain this? Probably yes. How many times have you updated Adobe Acrobat? How many times have you read through the information before saying approve? Probably never. How many people are actually going to read all of that information? I'm going to guess a rather low percentage. So they're going to come in, and who is going to have to explain it to them? Probably you guys. So this is already available. And so I think it's important to realize that. And in terms of what these diseases mean and how they present, because it's not always straightforward. Any question about any of that? Okay, so I presented genome-wide association studies. I presented it a little bit quickly because I was going to come back to it. Polymorphisms. Genome-wide association studies really rely on polymorphisms. So it's important to understand what a polymorphism is. And we talk about mutations and we talk about polymorphisms. And we sometimes use them interchangeably when we really shouldn't. There's an important distinction between these two. All polymorphisms are mutations. All mutations are not polymorphisms. So the difference is the frequency by which these occur. Polymorphisms are commonly occurring mutations. So if a mutation occurs in a population where at least 2 alleles appear with a frequency of greater than 1% in the population, it is a polymorphism. Mutations that cause diseases tend to be very, very rare. They don't come anywhere near approaching 1%, typically. So polymorphisms tend to be mutations that don't have much of an effect in terms of phenotype. They don't have a negative effect. They don't have a positive effect. If they had a negative effect, they would be much lower than 1%. If they had a very positive effect, they would increase in frequency in the population beyond 1%. So these are mutations that aren't really as critical in terms of our phenotype, is the basic idea. A commonly seen mutation, probably not causing a disease or a phenotype. So, lots of genomics utilizes SNPs. While they don't contribute to phenotype, they can be very, very valuable. They are oftentimes abbreviated as SNPs. So if you hear the term SNPs, that's what we're talking about, the single nucleotide polymorphisms. Single base pair variations, essentially point mutations, one nucleotide exchange for another. No insertion, no deletion, simply a GC base pair changing into a TA base pair, or an AT base pair changing into a GC base pair. And so here is one SNP where there's differences in the population. This person is homozygous for GC. This person is heterozygous, GC at one, AT at the other, and then this person homozygous for AT base pair. And then another SNP where essentially at a different location, but a single nucleotide base change difference. If you look at any two people, so if you turn and look at the person to your right or to your left, even though they are not your brother, sister, aunt, uncle, anything, you are 99.9% the same at the DNA level. Yeah. I always thought that was a fascinating thing. You talk about inbreeding. Humans are inbred. There's no doubt about it. 99.9% identical at the DNA level. 0.1% genetic difference that we have between any two people. But when you have 6 billion base pair, 0.1% corresponds to still millions of base pair differences because we have so much DNA. But we are remarkably similar at the genetic level. There have been about 1.8 million SNPs that have been characterized within the human genome, mapped to particular sites. And these can be really valuable. So a SNP map of each and every chromosome has been developed. Essentially where we can say, this is the SNP at this location, and that is the exact spot for that SNP. So what we can then do is map traits back. We saw that with genome-wide association studies. We can map them back based upon their linkage to particular SNPs. These SNPs occur approximately every 2,000 base pair along the human genome. So 2,000 base pair a SNP, 2,000 base pair a SNP, 2,000 base pair another SNP. This particular figure that you can barely see is chromosome 22, and it doesn't matter the details, but just that this is a SNP map for chromosome 22, and there's about 28,000 different SNPs that have been mapped back to chromosome 22, and we know exactly where each and every one of these is. So, SNPs. SNPs are rarely responsible for a disorder, and that should make sense to you. They are frequent in the population. If they cause a disorder, they should be infrequent. Essentially, selection, natural selection, should keep them pretty low. Or positive selection should keep them a whole lot higher. So generally speaking, they don't contribute a whole lot in terms of disorders. But they can be tightly linked to mutations that do cause disease. So we might have a deletion that is in a particular region and a SNP that is very close to that deletion. So later generations of people who inherit novel mutations will also inherit the same pattern of SNPs. In other words, in a particular region of DNA, you tend to inherit that as a block. So the SNPs go along with the mutation, generation after generation. What breaks them up is recombination that occurs during meiosis. So we have another term, which is very commonly used, called haplotype. And haplotype refers to the distribution of SNPs along a single chromosome. So what we're seeing here is, let's say that this is chromosome 8. And we're looking at different individuals. So this is one person, this is another person, another person, another person. And there are different arrangements of these SNPs. And each person may have a unique haplotype of chromosome 8. And it's the sequence of SNPs that are occurring. So these mutations that cause disease, they arise, but they arise very rarely in a population. So what will happen then is a mutation is going to arise on a particular haplotype. So let's call that the haplotype 2 background. So the mutation arose on the haplotype 2 background. When it's transmitted to future generations, that mutation will tend to be passed in the haplotype 2 background as well. And so this is where we can see this association. This mutation is associated with the haplotype 2. So if we are then screening individuals for this disease caused by this mutation, we'll find an increase in frequency in individuals who have a haplotype 2 genetic background. So the individuals have the disease, they have a SNP that's very close to it, and so we can identify those genes based upon the locations of these SNPs. And that's what we're trying to do here with these genome-wide association studies. So again, we have MS cases, we have controls. These are people who have the disease. And we are trying to say, for this particular SNP, is there an enrichment in those who have the disorder for this particular SNP versus the general population? There's problems with these. One of the problems with this is that we're assuming that all of the people here have the same disease for the same reason. And that may not be the case. Let's say that you wanted to do this with cancer. That was your disease. How many different types of cancer are there? Lots. So let's say you want to do this with schizophrenia. Are there different types of schizophrenia? We don't even know. Certainly possible. Different genetic basis for various forms of schizophrenia. Let's say that there's 15 different types of schizophrenia in the population. If you lump them all together, are you going to be able to identify a whole lot? Each group has a different pattern in terms of the genes that are contributing, so it'd be really hard to segregate it that way. So one of the things we have to try to do is really narrow it down. So instead of MS, this might be MS subtype 1, for example. Really specific, and then we're trying to narrow in on exactly what is the cause of MS1. We can still identify genes in this way, and hopefully then identify disease-causing or contributing diseases. So, pharmacogenetics and genomics. Pharmacogenetics, study of individual genetic variants that influence the response to a drug. So, individual genes. When we say genetics, like pharmacogenetics, we're talking about individual genes. Genomics is bigger, kind of big picture, 30,000-foot level, if you want to call it that. Looking at the genome as a whole. Assessment of multiple genes, if not the entire genome, simultaneously. The fundamental idea is avoid adverse drug reactions in individuals, and also best course of treatment for an individual based upon their genetics or their genomes. That essentially amounts to personalized medicine. Trying to do personalized medicine based on an individual's genetic information. So this strongly relates to SADRs, which are serious adverse drug reactions, SADR. About 15% of drugs approved in the US have the potential for serious adverse drug reactions. 100,000 people in the United States die every year from an SADR, and even with proper usage of the drug. This does not necessarily imply that the drug was used incorrectly. Many of these are where the drug was used exactly as it should be, but the individual still had a very serious drug reaction. So the question is, can we use genetics and genomics to try to get a handle on SADR? and potentially avoid these responses? And the early answer is yes, that we can. I'll give you a couple of examples of this. So, case first. 44-year-old, recently diagnosed HIV-positive patient is placed on the reverse transcriptase inhibitor, abacavir. So that's the drug that the person is put on. 2 weeks later, the patient comes back, presents as shown. And I don't think I gave you the answer on this in terms of your handout. Any idea in terms of the clinical presentation of this individual coming back 2 weeks after being placed on abacavir? No, not yet. I'm not surprised. I didn't expect you to necessarily know this. Students often get freaked out when they say, I should have known that. But I don't expect that you would know this. But in this particular case, the most likely syndrome that this individual is presenting with after being placed on abacavir is Stevens-Johnson syndrome. Have you been exposed to that? I'll give you a little bit of information. This is an autoimmune disorder, T-cell mediated damage to the skin, as well as mucous membranes. Causes death in about 10 to 30% of cases. So it is a very, very serious clinical presentation, Stevens-Johnson. Stevens-Johnson, after treatment with abacavir, has been linked to a particular HLA allele. On what chromosome? 6, right, chromosome 6. HLA 5701 is the particular HLA allele that has been associated with Stevens-Johnson syndrome after treatment with abacavir. Those individuals who have this allele have about a 50% chance of developing Stevens-Johnson. And if they do, they have a 10 to 30% chance of dying from it. Individuals who lack this allele have pretty close to a 0% chance of developing Stevens-Johnson. So there is a very strong association between having this particular allele and having risk for Stevens-Johnson. So the Infectious Disease Society of America has established that only individuals who are HLA-B 5701 negative should receive abacavir treatment. There are alternatives for people who have this particular allele, but they should never be given abacavir. So do we need to screen every single patient prior to administration of abacavir for this particular allele? That's a good question. And again, the genetics is not as simple and it's not as straightforward as we would like it to be. And the reason for this is that the allele varies in the population. Different populations have different frequencies of this particular allele. It is extremely low, for instance, in the Chinese population, 0 to 2%. Much higher in the Caucasian population, in the African American population, 2.5%, so pretty low. South Indian population, 5 to 20%, quite a bit of variation. The Thai population, 4 to 10%. And certainly other populations as well that we don't know yet. So it's not the same in every single population. So if it's almost unheard of for an individual who is of Chinese ancestry to have this mutation, should we screen for it? And essentially, due to the potentially lethal effects of abacavir, if you have this allele, the recommendation is yes, everybody should be tested for this. But this does have to be kept in mind. Is it more cost-effective to screen everyone or just those individuals who are at highest risk in the population? This is where things are not as straightforward as we would oftentimes like them to be in terms of what is the best approach. So there are a number of examples, and this is growing as time goes by. For instance, there's administration of codeine. And there are certain mutations, I'll talk about this in the next few, that influence codeine metabolism. There are several with respect to warfarin. Albuterol has a particular allele that it is associated with. Oral contraception and factor V Leiden, for example. Individuals who are factor V Leiden are at much greater risk of developing clots if they have this particular mutation. So identifying that mutation first can be valuable. The use of this is still being evaluated, and it is still controversial in terms of what is the best approach, and who do we screen and who do we not screen And can we afford to screen every single person for every single possibility? The answer is probably not. So we may have to be selective. Another example, cytochrome P450s. I think you've probably heard about cytochrome P450s, involved in drug metabolism. So for some drugs, it's involved in clearance, cytochromes, and others it's involved in activation of the drug. So if you're given a prodrug, you have to activate it. 56 different enzymes, 56 different genes, more than 90% metabolized by a set of 6. And this is the 6, with CYP CYP3A4 being the most important one. I'm not a pharmacologist. That's all I'm going to give you for that. I'm sure you've gotten or will get a lot more than that. But the CYP genes involved in drug metabolism. So there are a number of these that are highly polymorphic in the human population. Those polymorphisms correspond to proteins that have differing activities. So a number of these CYPs vary in the population in terms of the activity of the enzymes. So different combinations of alleles, different rates of drug metabolism as a result of these genetic differences. We can basically lump these into 3 broad categories. There are the poor metabolizers. There are the normal metabolizers. That's given the poor term, I think, extensive metabolizers, which I don't like that term at all. But that's the more common term, is extensive metabolizers. This is kind of intermediate. And then ultra-fast metabolizers. So those are the 3 kind of categories that we associate with drug metabolism. So this is a nice figure in terms of what can happen with respect to a drug if you fit into one of these different rates of metabolism. This is if the drug is inactivated by the CYP. So if the drug is inactivated by the CYP, then poor metabolizers, they inactivate slowly. So what that means is that the drug accumulates in the plasma in these individuals at a much more rapid level. And so with drug, we give the drug over time. Like take this drug every 8 hours, something like that. So a poor metabolizer, their concentrations go up and goes down slowly. And then they give it again, it ramps up. And so this blue area represents the therapeutic window for the patient. And so a poor metabolizer will shoot right past that and potentially to a lethal level. Whereas a normal metabolizer, you're in the therapeutic window for a long period of time. This is how then the dosage is determined, is this window. And then when you have ultra-fast metabolizers, they are very rapid, and so they will completely break down the drug before the second dose is given, and so have very little benefit to the drug at all. So ultra-fast metabolizers, very little benefit. Poor metabolizers, potentially toxic levels very rapidly. If the drug is activated by the CYP enzyme, it's kind of the exact opposite occurrence, where I've taken the same figure, but I've changed it around. So poor metabolizers will be very poor at activating the enzyme, or activating the drug. So they'll get very little benefit. Ultra-fast metabolizers will tend to activate very quickly. And so the drug will rise to very toxic levels right away. So it's different for ultra-fast versus poor metabolizers, whether the drug is activated by the CYP or it's inactivated by the CYP. And you have to keep those 2 categories in mind. The example, and I'll use this example and then I'll wrap things up. Morphine. Morphine is 10 times more potent as an analgesic than codeine. The benefits of codeine is that it's converted into morphine in the body. So codeine is what we give to the patient, and it has to be activated by the patient's enzymes. It is done by CYP2D6 enzyme. And for this particular enzyme, there are both ultra-fast genotypes and there are poor metabolizers at various frequencies in the population, and different amounts as well. So for instance, Native American population for CYP2D6, virtually unheard of. whereas in sub-Saharan Africa, about 3.4%. In the Caucasian population, about 7.2% are essentially poor metabolizers for this particular enzyme. So an ultra-fast genotype in this particular circumstance has the potential to be a lethal administration of codeine because it will rise much more rapidly than what we thought that it would. So this will be my last slide. No, I'm out of time. But this will be my last slide, I promise. 2012, FDA issued a warning on the use of codeine in children after tonsillectomy or adenectomy, and they said, essentially, use as little as possible. And on an as-needed basis, due to the fact that there are these ultra-fast metabolizers in the population, because kids were dying from what was typically a normal amount of morphine being used, or codeine being used. 2013, they kind of reversed themselves and said, no, better idea is don't use codeine at all in any children, period. And it is primarily because of these particular alleles that result in ultra-fast metabolizers for codeine. Okay, on time, almost. All right. Thank you. I'll see you guys tomorrow morning. By the way, I don't know where I have an office yet, but somewhere in one of these two buildings, I will be around.